So we're going to talk about the lytic life cycle first, and this life cycle is very similar to the animal virus life cycle that we talked about previously. So you'll see a lot of similarities, but I will point out some big differences that I want you to focus on. But overall, the order of the stages is quite similar. So the first stage is that adsorption or attachment stage. So that is when the virus is going to attach to the receptors on the host cell membrane. Now the primary difference here is the virus is a bacteriophage, and instead of using spikes, it is going to use its tail fibers at the bottom here, these little things that look like legs. It's going to use its tail fibers to attach to receptors on the bacterium. This is still a highly specific reaction. So this bacteriophage, its tail fibers are going to be able to bind to the receptors on a specific type of bacteria. So maybe this bacteriophage is specific for E. coli, but there would be another bacteriophage whose tail fibers would bind to the receptors for staph, or there would be another bacteriophage whose tail fibers would bind to the receptors for Bacillus anthracis that causes anthrax. But it's still a highly specific reaction where you have to have the appropriate tail fibers to attach to the appropriate receptors on the surface of the bacterium. Now, the penetration stage is quite different when we compare it between animal viruses and bacteriophages. So with animal viruses, when they were going to enter a host cell, there was two mechanisms of penetration: endocytosis or membrane fusion. But in both cases, the virus entered with a protective covering. It either still had its envelope on, still had its capsid on, or potentially even a capsid and the envelope. But at bare minimum, when the virus entered the host cell through endocytosis or membrane fusion, the genetic material was still encased inside of the capsid. And for that reason, we needed an uncoating stage to release the digestive enzymes, break down the capsid, and release the genetic material. It's not the case with bacteriophages. With bacteriophages, the virus injects only its DNA into the bacterial cell. So we can see here the DNA is being injected here in the blue. The rest of the virus, the icosahedral head, the helical neck, the tail fibers, they all stay outside of the cell and eventually are just disregarded. Only the bacteriophage DNA enters. Since only the DNA enters, none of the protective coverings, there is no need for an uncoating stage, which is one of the primary differences in regards to animal virus replication versus bacteriophage replication. The uncoating stage is not necessary for bacteriophages because only the DNA is injected. It's not encased in a capsid in this case, so there's nothing to uncoat. It's already essentially like pre-uncoated. So the uncoating stage is unnecessary and it's eliminated amongst bacteriophages. So eventually this bacteriophage DNA is going to basically hijack the bacterial cell. It's going to destroy the bacterial cell's DNA and manipulate the bacterial cell into producing thousands and thousands of more of additional bacteriophages. So in the next stage, we get to the synthesis stage. So in the synthesis stage, the bacterial cell, which is the host cell, is going to produce all the parts of the bacteriophage. So it's going to make thousands of copies of phage DNA for all the new bacteriophages. It's going to make all the different proteins that are needed to make up the bacteriophages. So it's going to make the little icosahedral heads. It's going to make the helical necks. It's going to make the tail fibers as well. So much like the synthesis stage in the animal viruses, in this stage for bacteriophages, the bacteria are simply just producing all the parts of a bacteriophage, including the DNA and the proteins needed to form the mature bacteriophages. In the maturation stage, or the assembly stage as we're calling it, it's the same thing as the assembly stage with animal viruses. If you make all the parts in the synthesis stage, in the assembly stage, you're going to put all the parts together. So at this point, we would start to assemble the mature bacteriophages. So really what happens is the genetic material is inserted into the icosahedral heads. The necks are then attached to the heads, and then the tail fibers are attached at the bottom. So basically, we are going to have thousands of mature bacteriophages inside of this bacterial cell. So that's essentially the same too, you're just kind of putting together different parts in different formation. The last stage is the release stage. This part's slightly different because with animal viruses, there was two different types of release depending on what type of virus you're talking about. Naked viruses would be released through cell lysis. Envelope viruses would be released through budding. With bacteriophages, there's only one option, and it's cell lysis. So eventually this bacterial cell is going to get so packed with bacteriophages that it's just going to rupture, so it's going to burst through cell lysis, and all these bacteriophages are going to be released where they will go bind to another bacterium and start the whole process all over again. But you could see where this would be potentially beneficial for the medical industry, because taking a tablet of these bacteriophages could allow them to infect bacteria and then ultimately destroy those bacteria. So that's why we think of the lytic life cycle as being something advantageous for us, because it can lead to the destruction of bacteria. This is what this would look like in real life. So here, this is E. coli, so we can kind of see the rod-shaped outline here. Now this E. coli is infected with a bacteriophage. And that bacteriophage, it's actually in the last stage, so it's in the lysis or release stage. And what's happening is E. coli is bursting through cell lysis. And all these darker areas here are all mature bacteriophages that are going to be released, where they will go bind using their tail fibers to new E. coli. And the process will just start all over again, and it will end in ultimately the death of E. coli. So that's kind of the good side of it. There is a dark side, and this is why there's still a lot of research to be done in regards to using bacteriophages in the medical industry. So there is the fear of this temperate life cycle, and again, in the temperate life cycle, you wouldn't kill the bacteria. Instead, you could potentially make the bacteria stronger. So in the temperate life cycle, it kind of starts out the same. The bacteriophage is going to bind to the bacteria using its tail fibers. The bacteriophage is going to inject its DNA into the bacterial cell. No need for uncoating. And then this is where it kind of shifts. So before, in the lytic life cycle, this bacteriophage DNA would essentially take over the host cell. It would destroy the bacterial cell's DNA, and then it would manipulate the bacterial cell into producing thousands of new bacteriophages, and then eventually the bacterial cell would burst and release all those bacteriophages. Well, that's not happening here. Instead, the bacteriophage DNA is actually going to incorporate itself into the DNA of the bacterium. So the bacteria is going to stay alive and well. It's not affected. It now just has some extra genetic material. So it has all its, original bacterial DNA, but now it has this little extra piece that originated from the virus. So every time this bacterial cell divides and makes more bacterial cells, so this bacterial is going to divide and make 2 more, and then these 2 would eventually divide and make 4 more, etc., etc. But every time this bacterial cell divides, the DNA is going to get passed down, and that DNA is going to include the viral region. So whatever was in this DNA could be potentially advantageous for the bacteria. Sometimes the viral DNA didn't have anything particularly advantageous. Maybe it gives the bacterial cell the ability to be able to sustain a certain temperature, or maybe produce a flagella. Nothing too serious. Sometimes, though, the viral DNA can tell the bacteria how to produce a dangerous toxin, or potentially how to become resistant to an antibiotic, things that are much more dangerous. So when that happens, and the bacteria learn how to do something new, that is called lysogenic conversion. So when a bacterium acquires the ability to, acquires a new trait, such as the ability to produce a toxin or become resistant to an antibiotic, when it develops this ability to do something new after becoming infected with the bacteriophage, that is called lysogenic conversion. I have two examples here, Vibrio cholera and Clostridium botulinum. Both of these bacteria cause very dangerous diseases. Vibrio cholera causes a disease that we know as cholera, which can lead to life-threatening dehydration. Clostridium botulinum can lead to full-body paralysis within a matter of hours that can be deadly. So these are really dangerous bacteria. But they weren't always dangerous. At one point, these bacteria were non-pathogenic bacteria found in the soil. They couldn't do anything to you. But what happened is at one point in history, these bacteria got infected with a bacteriophage. And that bacteriophage went through the temperate life cycle. And it inserted its DNA into the DNA of these bacteria. And then when these bacteria, went through division and made more of their own, those future bacteria also had that viral DNA that came from the bacteriophage. And what that viral DNA taught them to do was make very dangerous toxins. So they went through lysogenic conversion, and they went from just being normal, non-pathogenic bacteria to now all of a sudden being very pathogenic, very dangerous bacteria that are able to produce life-threatening toxins. The cholera toxin induces vomiting and diarrhea, and like I said, could cause someone to become severely dehydrated within a matter of hours. The Clostridium botulinum bacteria, it is able to produce the botulinum toxin, which can lead to full-body paralysis. So choose the right bacteriophage, and it goes through a lytic life cycle, and we kill bacteria. Very good. Choose the wrong bacteriophage, and it actually infects the bacteria, goes into a temperate cycle, and incorporates its DNA into the bacteria, which could potentially teach the bacteria how to do something new, which we would call lysogenic conversion. Now it's able to do something new, such as produce a toxin, but again, it could even be something such as becoming resistant to a specific antibiotic, being able to survive in a certain type of an environment that it wasn't normally able to. Maybe it developed an appendage that it didn't typically have, like a fimbriae or pili or even flagella, something like that. So there are obviously a lot of situations in which we would want to study viruses. So often we are culturing viruses in order to study them in the lab. That's important when we're wanting to develop vaccines, when we're wanting to develop antiviral drugs, which basically are medications that stop the virus replication cycle. If you can stop a virus from attaching to a host cell, you can stop its replication cycle so the infection won't progress. And then we obviously study viruses for diagnostic purposes to see if an individual is infected with a particular virus. So there's a lot of situations in which it would be beneficial to study a virus in a lab. So the next thing we're going to talk about are the various different routes for studying viruses.